Bill Kaelin, welcome to this interview.
Thank you.
It's entitled Reflections on a Life in Science. Is it possible that we could have been having a different conversation with you, perhaps? Reflections on a life in, I don't know, law, life in arts. Were you always going to be a scientist, or was there another possibility?
Well, thank you, and good morning to everybody. I wish I could be there with you in person. I'm as surprised by the path that I wound up taking as anyone could possibly be. First of all, I would say as a young person, I was a rather undistinguished student, but my saving grace was that mathematics came easily to me. And because it came easily to me, I could get good grades without actually studying, which was my kind of subject. I didn't like the subjects where I had to study, particularly the ones that involved a lot of rote memorization, but I liked mathematics quite a bit. And then in the mid-70s, when I was in high school, computers came along and I became intrigued by computers and computer science. And so initially I was thinking maybe that would be the direction I would go, because computers to me seemed like math come to life. A lot of the reasoning and logic of computer science to me had similarities to mathematics. But now comes the embarrassing part. I simply did not see the personal computer revolution that was around the corner. And I wasn't sure whether I could make a viable career in computer science, or if I did, I imagined I would be in a dark room punching computer cards, working for some big company or perhaps the military, writing computer code. And I wanted a job where I would be interacting with people every day and where I felt like I could do some good in the world. And so I thought maybe I should start segueing towards medicine. And so I trained as a clinician, both in internal medicine and medical oncology. And in fact, my limited laboratory experience in college was so bad that my professor gave me a nearly failing grade, and actually wrote in the margin of my transcript that Mr. Kaelin appears to be a bright young man whose future will lie outside of the laboratory. So as a 17 or 18-year-old, I did not think of this as a very auspicious start to a laboratory career. But fortunately, when I was a postdoctoral fellow studying cancer medicine, I had the opportunity to work in a laboratory. And by now, and this is now the late 80s, early 90s, the molecular biology revolution was really taking hold. And I had a wonderful mentor and a wonderful project to work on, a great laboratory. And I was having so much fun, partly because my expectations were so low that I easily exceeded them. And then I started to get some success and a little bit of what we call a halo, where people decided maybe I could do this after all. And so I decided, as long as I was having fun and felt like I was making a contribution and getting paid for it, I'd continue doing science. Otherwise, I'd go back to being a clinical doctor. And that was 30 years ago.
Okay. So the halo effect seems to have lasted until now.
So far, thankfully.
I think you know what we can do. I think we can take a quick peek into the auditorium in Barcelona and just wave hello to our audience there. So I wonder if we can pull up the auditorium view of the Institute for Global Health. Is that possible that we can take a peek? I just want to wave hello.
Yeah.
It's not appearing yet. No, we're being too ambitious. I'm so excited by this hybrid that we're able to achieve, where we're speaking both to the audience there and to the online audience. But no, it's not coming. If it pops up, we'll wave. That's fine. Okay. When you were giving your lecture on Wednesday, you mentioned something that was, I think, very interesting. You talked about having a good nose for a problem. Yes. And of course, one of the things that I suppose people are hoping to get from listening to you today, talking about your life in science, are the keys to the success.
Yes.
How you live a successful life in science. I mean, I suppose if you live an unsuccessful life in science, you probably move and do something else. But how do you make a successful life in science work? And one of them is obvious. One of the keys to success is obviously being able to identify the right problems to ask. How would you define this idea of a good nose for a problem?
Thank you. I do agree. This is so, I think, fundamentally important. I've heard a number of scientists remark on this over the years. I've heard one person say, it's as hard to work on an uninteresting problem as it is to work on an interesting problem. And there's some truth to that. Then there's an opportunity cost to everything you do. If you're working on X, you can't necessarily have enough time and resources to work on Y. You can tell my mathematical training now. So I think what you work on is fundamental to your eventual success. And as you say, I do think a lot of it comes down to having instincts or intuition regarding what is a good problem to work on, as opposed to something that's likely to be a bit derivative, tangential, unlikely to change the way people fundamentally think about a problem. And I say instincts and intuition because to this day, I don't know how much of this is nature versus nurture, meaning I don't know to what extent scientists are born with it, perhaps, versus having it imprinted upon them by other great scientists. I think there's certainly a component of the latter. If you're around great thinkers and great scientists and you see the way they think and you see the way they tackle problems, I think it has a profound influence on you. In fact, long before I won the Nobel Prize, I can tell you that I was struck by the fact that every Nobel laureate I had ever listened to, and I had the privilege of listening to quite a few, had this property that they could be at a lecture that was actually outside their field of interest, and yet during the question and answer period, they would almost always ask the singular, most penetrating question that was raised by that lecture, and they would not allow themselves to get distracted by the things that were sort of off at the periphery. So maybe you can think of that as seeing the forest for the trees. So I think one key is to surround yourself by, if you can, great scientists, great thinkers, and I think some of it will rub off on you, that you'll develop instincts. But it's a great question. If you go backwards in time and look at some of the initial observations that eventually led to Nobel Prizes, I'm struck by the fact that they were often just head-scratching curiosities, but someone had the instincts to say, I think there's something here. I think if I could understand this, maybe eventually, but I think it kind of goes back to, why do you do science in the first place? I think most good scientists just like solving interesting puzzles, and why certain people find certain things interesting and others not, I think is a great mystery. But I guess the last thing I'll say is, because I was a clinical doctor, I was giving up a lot to become a scientist. And so one of the things I demanded of myself is I had to at least believe that when I look back on my career, that I wouldn't be caught in the trap where I would say, everything I did competently and well, but it all would have been done with or without me. And so we've tried on occasion to take risks, take chances. And then I would also say I had the benefit of my clinical training, which I think helped guide me to certain clinical conundrums that could then be tackled in the laboratory. And that turned out to be very fruitful.
Gosh, there was an awful lot in that answer to dissect. Now we're beginning to get lovely questions coming in, and actually I forgot to say, do please tell us where you're writing from, whether you're writing from the auditorium or writing from somewhere else, and if you'd like to tell us where in the world you are, that's always lovely. And I'm also getting a note to say we can't see the auditorium, unfortunately. What a pity. Never mind, we wave to you anyway, and wish you could wave back to us. But Sober asks about the skills required to make progress. And I guess you've already been touching on that, this idea of being around people who are asking good questions and learning from them, and logic. Let's just focus on logic for a second, because you've mentioned it a couple of times. Your mathematical ability gives you, I suppose, a handle on that. But is that something that is essential? Do you think a young scientist has to be good at logical thinking?
I firmly think that's the case. But let's dive a little bit deeper, because the question, I think, is what are the ingredients and what do you need to master?
Mm.
I've always been struck by some of the ways I've been wrong over the years. I thought that the hardest part of science would be learning the techniques of science. That actually turns out to be, in my view, the easiest part of science. I think I've yet to see a technique that, if properly explained by the right person, I couldn't begin to master. In fact, in many cases, once it's explained to me properly, I'm sort of like when a magician shows you how they do their tricks, and you want to say, "Oh, that's all it was." It seems so off-putting until someone actually explained to me how you actually did it. So I would say I would not focus on the technical aspects of science, although yes, you have to over time acquire some technical skills, but that's the easy part. The harder part is, I think, learning to think clearly and logically, framing a question in a way that then leads to hopefully a very powerful experimental design that will answer the question, or hopefully answer the question. And I've sometimes made the comment that technicians do experiments and scientists design experiments that have meaning, because they include the right positive and negative controls, so you can actually interpret the results. Just getting results per se is not the name of the game. The name of the game is to get results that actually teach you something, and that usually is linked to the power of the controls, and that's where we get now into the science and into the logic and reasoning part of science. And so that's another reason why I think you want to be hopefully surrounded by great thinkers, great scientists, because they can help you in this regard.
Mm. And I'm intrigued by this thought of you wanted to have done something that you made a difference to, that others would not have done. So that means you really have to know yourself. You have to know what you're good at and see where you can make a difference.
Yeah, I think that's right. Now, I've also been struck how many times we thought we were doing something that was truly unique, only to discover that we had someone hot on our heels. But that's okay. That's the way science works. In fact, sometimes independent corroboration, independent but simultaneous corroboration, is a very good thing for the field, as happened with the work that led to the Nobel Prize, where Sir Peter Ratcliffe and our group effectively simultaneously discovered that HIF was prolyl hydroxylated. But another sort of way that I try to approach this problem is to say to myself, is the next experiment so obvious that it's going to get done well with or without me? Because if the answer is it's certainly going to be done with or without us, I have to really say, then why are we doing it? Now, sometimes we do that experiment because we need that piece of the puzzle to get to what we think is going to be the even more exciting piece of the puzzle, or we rationalize that we can do it better or more quickly than anyone else because we're perfectly poised to do the experiment. But there have been times we've just walked away from obvious experiments that would have been publishable, would have been fundable, but it was very clear that 10 labs were going to jump in and get the answer, and at that point it became uninteresting to us, or at least getting back to the opportunity cost, we thought it would be more important for us to try to devote our attention elsewhere.
That's absolutely fascinating. And a lovely question has come in from Geordie Sunya. We're talking about success in science, but what makes a scientist successful? How do you define success?
Oh, this is such a great question. Thank you. Well, let's start with how I would not define success in science, and this may sound paradoxical coming from me. To me, it's not measured in prizes. It's not measured in which prestigious societies you've been elected to. It's not measured by how many papers you've published. It's not measured by your citation factors or impact factors per se. I think, first of all, success in science is, are you doing something that you enjoy so much that it feels like you're playing rather than working, which is the way I feel when I go to work. I mean, when I was a clinician, I had tremendous respect for clinical medicine, but I didn't feel like I was playing. I felt like I was working, and I didn't feel guilty that people paid me to do it. Whereas I feel as a scientist, I have this great privilege of coming to work each day and again, solving puzzles and being surrounded by brilliant people, and on occasion, understanding something that's never been understood before. So it's that joy and satisfaction to me that begins to mark what it is to be a successful scientist. I certainly think it has to be emphasized that to be scientists, we want to do work that in the end is measured by whether other people could replicate the findings, whether the findings were true, and whether they were sufficiently robust that other people could build upon them. And so I think that's how you begin to measure success. And the good news here is I think if you focus on those things, doing your science well, asking good questions, focusing on seeking the truth, if you're really lucky, those other things will fall in place, namely the papers and the recognitions, and maybe if you're very, very lucky, the occasional prize. Whereas I think if you just focus on the prizes and the accolades, you may wind up a very bitter, unhappy person, because there are things that are beyond our control that go into winning such recognitions.
Mm, mm, mm. Were you surprised by how important your work turned out to be?
This may sound odd, but not completely, because getting back to the importance of what you work on, in 1993, when we decided to work on the von Hippel-Lindau gene, I knew from my clinical training the clinical features of individuals who are born with a defective version of the VHL gene, so-called von Hippel-Lindau disease. And so I knew from those clinical features that if we could understand the functions of the VHL protein, we would certainly learn something about how the body regulates angiogenesis, which was of great interest in the 90s, because there was a thought, which turned out to be at least partly true, that you could inhibit tumor growth by blocking angiogenesis. I also knew that the VHL gene was going to be a critical player in kidney cancer, which is one of the 10 most common cancers in the world. So the hope was, if we really understood what the VHL gene did, eventually this might lead to new treatments for kidney cancer, and that turned out to be the case. And then finally, again, based on these peculiar clinical features, it was pretty clear to me that the VHL gene product would play some important role in oxygen sensing. And again, I knew from my clinical training that oxygen plays an important role in many diseases, not just cancer. So in the back of my mind was the hope that maybe one day this would be impactful. But we also knew there are no shortcuts. I mean, everyone wants to translate. Everyone wants to cure this or cure that. But you have to roll up your sleeves and generate the knowledge first. And so I didn't know whether, I guess the part that was an unknown was whether this was going to take 1 year, 10 years, or 100 years. So I'm happy I lived to see at least some of it reach a point where it could be translated.
Thank you. A nice question has come in from Hernando del Portillo from IS Global, who is watching online. And he wants to know how the advent of machine learning, AI, the enormous quantities of data are going to change science, and I suppose in particular whether they're going to speed things up.
Well, I would say this. First of all, when you pick an area in science, again, why different people find different things interesting, I don't know. What you have to find is what's interesting to you and what you're passionate about. And for some people, that's going to be computational biology, AI, machine learning. And there's no doubt that these are very powerful tools that will teach us things. Having said that, let me sort of artificially dichotomize what's actually a continuum. So at the one end is what some people have referred to as the reductionist biologists. These are the people who want to go deep into a problem. They want to understand mechanism. And frankly, a lot of reductionist biologists, including myself, are what sociologists would refer to as sort of the lone hunter-gatherers who just follow their curiosity, look under rocks, study their gene, their protein, their pathway, but they go very deep. At the other extreme now are what people call the systems biologists. These are sometimes the people who do these large omic data sets, large-scale DNA sequencing, proteomics, now single-cell RNA-seq. They generate these incredibly dense, rich data sets, but often they're, in my view, they're quite descriptive of a system, but they don't always tell me how something works. And I want to understand how something actually works. So I think the two approaches to science are complementary. And as I said, I'm sort of artificially dividing into two, what's actually a continuum. But my concern for the young people is that a lot of the technologies that are currently employed by systems biologists are very trendy and sexy, you might even say, and they're often the shiny new thing. And I would sometimes pause before just following the next shiny new technology, because first of all, sometimes they go in and out of favor. And when not used properly, I've sometimes referred to some of these experiments as sort of the molecular equivalent of the blind man and the elephant, if people remember the story of the blind man and the elephant. So yes, you've measured, tens of thousands, maybe millions of analytes, and you tell me which went up and down in this particular condition. But again, I just want to know mechanistically how things work. The great geneticist Seymour Benzer once said, a good experiment should be pretty and witty. And so I like experiments that are pretty and witty, and I say, boy, that person was really clever to come up with that way of asking this question, and now I understand something. So again, I'm in the camp that I think machine learning and AI is certainly here to stay, and will be very complementary to maybe the people like me who try to go a bit deeper.
There's a lovely question. I'm going to turn to COVID in a second with a question from Rafa, but just before that, on the pretty and witty, on Wednesday, you also said, I remember, a good experiment is a simple experiment.
Yes. Well, I caution students, I think, and I fell into this trap when I was starting out also, that you thought that the best experiments were the most complicated experiments, the most sophisticated experiments, the ones that had the most bells and whistles, if you will. But of course, the more complex the experiment, the more things that can go wrong. So I always tell people, simple is not bad, simple is good. The simplest experiment that answers the question clearly is the best experiment.
Mm-hmm. Thank you. So much to talk about with COVID. Rafa Villaswan asks how you think the COVID pandemic is going to influence the future of research, medical research.
Well, I think one thing it's done, and this meeting is a good illustration, it's opened our eyes to the ways we can communicate with one another, share ideas, share data. I think that's very important. So I think moving forward, hopefully, scientific meetings will be much more accessible to people around the world than they ever were before. I think that's a wonderful thing. I think we have to be a little cautious, though. A lot of people, especially in the lay public, I think are trying to extrapolate from the COVID experience to other diseases, in some cases, I would say a bit unfairly. So as what I pointed out, the reason we could move so quickly on COVID was the COVID problem had largely been reduced to an engineering problem as opposed to a scientific problem, in the sense that, thankfully, because of decades of investment in basic virology and immunology and protein science and nucleic acid biology, when COVID came along, we weren't starting from scratch. You could start to make pretty good guesses about what each gene was going to do. You could make pretty good guesses as to which would encode proteins on the cell surface and hence be the best targets for potential vaccines. Thankfully, the breakthroughs in nucleic acid biology came along just in the nick of time, appropriately being recognized now around the world, including with this year's Lasker Prize, I think. The work that led to mRNA vaccines really it made it almost a plug and play situation. So now it was really an engineering feat. How could you now scale this? How could you make the vaccine rapidly at scale and test it quickly? And then in my world of cancer, of course, one of the big differences is that a virus has on the order of, say, 10 genes, whereas a human cell has over 10,000 genes. And we know in cancer, it's a multi-gene problem where in any given cancer, there are multiple genes that have been mutated. So people will ask me, well, now, of course, we're going to cure cancer in the next year or two based on what we learned from COVID. I think that's a gross oversimplification, but there are learnings there in terms of sharing information and just the power of investing in new knowledge and having it at your fingertips when you need it.
Thank you very much indeed. And on the first point of how there's a kind of democratization going on through what we've learned about how we can share with each other digitally and meet each other digitally, Pablo asks whether you miss traveling.
Well, I certainly miss traveling to places as beautiful as Spain. I've enjoyed my trips to Spain very much over time. So I do and I don't. I would be the first to admit that on one level it's inefficient, but it's so pleasurable. And as I think every scientist would admit, sometimes the most important interactions occurred in the bar after the seminar and not necessarily in the auditorium. So, and I can think of several times where chance meetings I had at meetings, I'm thinking about one in particular in Paris about 20 years ago, where I first met one of Sir Peter Ratcliffe's scientists, that was quite pivotal, where we exchanged some information offline that turned out to help both laboratories. So I do miss the travel, but I also understand that it maybe isn't the most efficient way of communicating.
If the world opened up again magically in 22 and everyone started traveling again, would you go back to your old pattern, or do you think you'll change?
I think I'll do somewhere halfway in between. I think I will do some traveling, partly for the reasons I mentioned. I do think that face-to-face contact is important, and I find it very pleasurable to, of course, go to beautiful places around the world. But I'm certainly not going to be making 3 or 4 trips a month as I was once doing.
It's a radical change. Okay, let's switch direction. Lionel Guélon asks who drives science these days? Is it the researchers asking the questions or the funding bodies paying for them to do so?
Oh, that's a very important and astute question. I'm fearful now that the funders have a little bit too much influence on the directions that science takes. And it sort of goes back to what we were saying a moment ago. Of course, everybody likes to dream about doing translational work that touches patients or improves the lives of people in some way. So everyone has, I think, that goal. But as I said earlier, there are no real shortcuts. So real translation doesn't happen when you put a gun to some young scientist's head and tell them, well, you're only going to get funded if in 5 years you can promise me that you're going to cure toenail cancer, or make up something. And by the way, we want to see your work plans, and you got to tell us what you're going to be doing in years 3, 4, and 5. And if you're not doing those things, then you're not going to continue to get funding. You know, that's the way you manage, again, an engineering problem. An engineering problem, it's reasonable to say, this is the goal, e.g., we're going to put a man on the moon. It's going to take us about 10 years. And each year we all have to agree together that this is what we're going to do towards that goal. That's how engineering works. Science, as you know, doesn't work that way. I always say, if you actually are doing in year 4 or 5 of your grant what you said you would be doing in year 4 or 5 in your grant, you're either doing something quite uninteresting and boring, or you've had blinders either put on you or that you put on yourself that's preventing you from following the science where it actually wants to take you. But time and time again, it's following where the science that takes you that actually leads to the big transformative discoveries that change the world. Think of CRISPR gene editing as an example. So I think we just have to acknowledge that when we're doing science, and now I'm speaking primarily to policymakers and funders, the only thing you can say for sure is the more you invest in the generation of knowledge, the faster we will arrive at places where on occasion that knowledge is actually useful. And hence we'll be able to improve the lives of people. But you can't, you know, I see this all the time where people are sort of held to these timelines and deliverables that are completely unrealistic. So I think one thing that I think made American biomedical research so strong beginning in the middle of the last century was what I think was a very wise decision, and I understand it actually was contentious at the time, but the wise decision to have the public sector, namely primarily the federal government, invest in basic and fundamental research because it was so open-ended and the timelines and deliverables were so unpredictable that the private sector wouldn't invest in it. But then to say, let the private sector be the harvesters. So we'll do the pump priming with public funds, we'll create new knowledge, and then on occasion, some of that knowledge will be useful, and then the private sector can step in and do the translational work. And I think that formula served us very well. So I think it was, whereas I fear in some countries over time, they became a bit too short-sighted with funding in terms of, well, what are you going to do for me in 5 years? And that's not really the way science works.
Sure. It's a question of trust, isn't it, really?
Yes. It is a question of trust. And there's a very important word that comes to mind when you say trust, because sometimes I tell people, I don't think we should micromanage what scientists do with their resources any more than you would micromanage what a great artist was doing. But that doesn't mean you can't hold them accountable. So I think you can say after 5 years, what did you do with the funds and the resources? And if you say, well, we did the following experiments, we did them well, they didn't turn out the way we expected, but then this one thing happened and we went in this direction, that's okay. Whereas if you just went and were sipping gin and tonics in Mallorca, then that's a different problem.
Which must presumably become a danger when you become a Nobel laureate and people invite you to New York.
I think so far I can avoid it.
Okay, Georgie Law, who's tuning in from the UK, asked a question about the future of scientific education and whether it is changing, whether it needs to change. How do you think the future of scientific education is looking?
Well, I'm embarrassed to say, and I do mean this, that I'm unfortunately pretty far removed from the way science is being taught these days, but I am, now that I'm a Nobel laureate, rethinking that position. And so, for example, I'm trying to think of ways that I can become helpful in terms of scientific education in the United States, especially in urban centers in the United States with underrepresented minorities who didn't have some of the benefits I had growing up. So I'm very interested in this topic of how we teach science. I mentioned the other day when I spoke, I actually found biology, frankly, pretty boring in high school and college because it was so descriptive and so taxonomic. It just seemed like stamp collecting. And it was a lot of rote memorization, something I detested. So I think it's really important that we teach science in a way that's engaging intellectually and gets people excited. I think, of course, now biology has matured quite a bit. So I'm hoping the better biology teachers take advantage of that to really bring the subject to life.
Thank you very much indeed. The questions are pouring in now, so lovely. The question of diversity, reaching people in in deprived areas you were just talking about, but in general, introducing more diversity into science.
Yes.
How important is it that that happens, do you think, for the good of science?
Well, this has been studied even beyond science. I think diversity just in general is helpful in almost any human endeavor, because we all come with different experiences, different skill sets, different perspectives. And so, for example, now that I'm a little bit older, I serve on a corporate board of directors, and people have studied this, and corporate boards that are diverse consistently outperform corporate boards that are sort of monochromatic, let's put it that way. So I think diversity is very helpful, and it's especially helpful, I suspect, in science, because again, what makes science work? Science works by challenging your biases and challenging dogma. And what better way to do that than to bring in people with, again, lots of different backgrounds and different perspectives.
Thank you very much indeed. Thank you. Okay, a question that I imagine is close to your heart. Fidanka Vasileva, who's joining from the Biomedical Research Institute of Girona, wants to know whether it's possible to be a clinician and a scientist at the same time.
Yes, this is a question near and dear to my heart. So first of all, I don't think it's one size fits all. I don't mean to prescribe the right answer. In fact, sometimes when I'm asked for advice, I preface my remarks by saying that if anyone who's reasonably well-adjusted has probably already rationalized why what they did was the right thing for them to have done. And that's a healthy thing. But what's not so healthy is when they project that onto everybody else and say, well, this is what you should therefore do with your life. So this is a very personal decision. So let me just give you a couple questions you might ask yourself moving forward. So when I was a clinical doctor, I loved, as I said, clinical medicine. I think I was actually a pretty good clinical doctor. So as I began to do science, one thing I said to myself was, if the choice is to be an A clinical doctor versus a B scientist, I'm now using the American grading system, it would make much more sense for me to be an A clinical doctor, because I suspect the world already has quite a few scientists who are Bs and not As. So that had some implications, however. It meant I had to really get the training and spend the time necessary to hopefully become an A scientist rather than a B scientist. And then I've already mentioned a moment ago that I wanted to reach a point where if I was going to give up doing clinical medicine, I had to think I had the chance of doing something that wouldn't be done with or without me scientifically, or at least wouldn't be done quite as well, that I was really going to have an impact, as opposed to being just one more person sort of laying bricks in the wall. Now, the reason I tell you that is eventually that meant, at least for me, that I couldn't spend 50% of my time in the clinic and 50% of the time in the laboratory. It was hard enough working virtually full-time in the laboratory to sort of compete, if you will, with the very best scientists in the world. And I used compete there only to say that I had heroes and role models that I was aspiring to be like. And so for me, eventually, I segued towards being a full-time scientist, because otherwise I think you run the risk of being what they would say, neither fish nor fowl. Now, for some people, they're willing to make that bargain, that they're a reasonably good clinical doctor and they're a reasonably good scientist, but that to me wasn't a bargain I was willing to make. I wanted to feel like there was a chance I could do something special. The final thing that helped me, getting back to puzzle solving, is a wise professor once told me, common things are common. And so in clinical medicine, like everything else, there were many things that were interesting the first or second time I saw them, but they weren't nearly as interesting the 30th time I had seen them. So that made it a little bit easier for me to start to segue towards science, because again, some of the thrill of my early clinical medical years was starting to go away, simply because I had seen some of these stories so many times before and I knew how they ended.
Thank you very much indeed. Of course, among Nobel laureates, there are some very strong advocates of the physician-scientist role. It's interesting.
Absolutely. Well, thank you, because the other part of that question, which I'm sometimes asked, or I should say I'm sometimes asked the question in the following way: if you knew you were going to be a scientist, would you have gotten all of that clinical training? And was that clinical training actually useful? And here, I think the answer is a resounding yes and yes. I would have done it again in a heartbeat, because again, any year where you're learning things and you're having fun and you're getting paid to do it is a good year. And the early years of my clinical training were wonderful. But more importantly, I see many scientific problems through the eyes of a clinician. And so I see some of the opportunities. I also, as I said earlier, there's no shortage of conundrums that arise in clinical medicine that are just ripe for understanding through laboratory-based research.
And it loops back to that earlier answer of really the feeling for the importance of the problem, the nose.
Yes.
So Emmanuel, who's joining from the library of the Faculty of Medicine in Barcelona, wants to know what advice you have for young scientists who are trying to choose their research field.
Well, I think it depends a little bit upon the stage, but if you're still in training, I would say what you're working on is maybe question 2. Question 1 is, who am I going to be working for? In what laboratory? Is this a laboratory that really does a good job teaching people the art of science, getting back to our earlier conversation, teaches people how to frame good questions, teaches people how to think clearly, logically, design wonderful experiments, generates data that's unambiguous, comes up with those occasional pretty and witty experiments we talked about. One thing that we've gotten away from, unfortunately, a little bit is, it used to be, and I've written about this, that papers had fewer claims, but more lines of evidence to support those claims. And the scientists I admired came up with non-obvious, sometimes very clever experiments that kind of corroborated the findings from the initial set of experiments that were maybe more pedestrian or prosaic. So I think I would first ask, who's my mentor going to be? I had heard, Adam, maybe you can tell me this, whether this is true or not, but when I was a young trainee, I heard that one of the best predictors for winning a Nobel Prize was having trained for a Nobel laureate. Like most jokes, I think there's a little bit of truth actually to that, but the sentiment is certainly correct. I think you want to train with somebody who's really a terrific scientist, who will teach you the arts and skill of being a scientist. I hope you find the questions they're working on interesting. That's important, but I think that's almost a secondary concern, because frankly, when you start your own laboratory, you're probably going to be working on something entirely different anyway. But if you've been taught how to think like a scientist and how to break down a question into a set of experiments that will yield answers, then the world's your oyster, and then it's later up to you what you want to work on. If they're referring to that they're now an independent investigator, all I can say is, I hope you've had some of that imprinting along the way that we talked about, and you have some instincts, but I can't tell you what to find interesting. That's something that only you can answer.
Okay, and the last question that's really related to science, from Julia Polarolo from IS Global, watching again online. What's your advice to scientists who are just burdened with negative results?
Yeah, before we answer that, I'll give to the last question, one last litmus test. I sometimes say, go into a closet, close the door, and say out loud what question you want to answer. And in my view, it should almost sound a little bit bold and a little bit audacious, but it certainly shouldn't sound pedestrian. And so it shouldn't sound like, well, I want to find the 5th SP1 binding site in the uninteresting gene promoter. It should be a little bit higher altitude than that. And it should almost sound a little bold and a little audacious. And I will say, one of the things I've learned now being at Harvard for many years, and I should tell the students, I was rejected from Harvard University for college. I was rejected from Harvard Medical School for medical school. And I was rejected from the Harvard hospitals for my residency. So in baseball, that would be three strikes. So it took me a while to get here. But what I have learned is one of the valuable things of going to a great place like a Harvard. I used to think it was the professors and the mentors and the fellow students, and all that's true, but you can find that other places. But I think when you train at a good place, you become a little bit more audacious and you're willing to ask bold questions because you kind of have this sense of entitlement. So I think if you can get the right balance of self-confidence and self-doubt, you can move forward and ask big questions. So now we're talking about frustrations, though, in science. So I mentioned this the other day when I was asked a similar question. So I think the first thing to do is I don't let anything that works go unnoticed and uncelebrated, precisely because there are so many failures. So I don't take anything for granted in the lab if it's worked, even if it's some relatively minor, trivial thing, you know. And I said when I was a postdoc, even cutting a plasmid with a restriction enzyme and getting the right number of bands, I would be doing fist bumps because to me that was magic. But I always tell people, first of all, take some joy in the little things that do work along the way. Second, recognize that even the things that quote unquote don't work almost always are getting you one step closer to success. I'm sometimes been struck how many times we had to do the experiment that in hindsight was destined to fail to finally have the light bulb go off and get to the experiment that was going to solve the problem. We just needed to see the failure first to really force us to think a little bit better about what it was we were doing. And you'd be standing in the shower going, oh, I'm so stupid. Well, of course that was never going to work, but now I can see what was going to work. So even failures can be quite informative. I think the tricky part, however, and this is really important, and this is probably one of the most important jobs of a good mentor, is it is clearly the case that sometimes you can be going along doing experiments, and to use an oil drilling metaphor, it's just becoming clear this is a dry well. There simply is no oil. You can keep drilling, there's no oil. And so I think that's when a good mentor has to step in and say, you know, you're too good to fail, and if you fail, it's really on me. We better plant you in a more fertile area because, you know, this just doesn't look like it's going to be fruitful. But that can be hard because sometimes, you know, you've invested a lot of time and effort and you think, oh, if I only drill another 10 feet, I'll hit oil. But sometimes there just isn't any oil.
Thank you very much indeed. Okay, we have to draw it to a close. You've worked so hard answering all these lovely questions. But I just wanted to finish on the Nobel Prize because people do ask about that. And for instance, combining questions from Alba and Melissa, did you, just very briefly, did you imagine receiving the Nobel Prize? And when it came, how has it changed life?
Well, so I think that's a very important question. So firstly, when we saw that the HIF protein was prolohydroxylated and started to really see this molecular circuit come into focus that was involved in oxygen sensing, you know, we knew that that was the kind of discovery that sometimes led to Nobel Prizes. But I should point out to the students that had the mechanism not been beautiful, not been elegant, been a variation of something we had seen many times before, no one would have been discussing Nobel Prizes. So that's one of several places where, as I said, luck really comes into play. I mean, this was a tribute to nature, not us, that the mechanism was so elegant and unexpected. As one laureate once said, most Nobel laureates really, if you look at it, are more about the discovery than the discoverers. But yes, we did know that we had been fortunate to have discovered something that was beautiful and elegant and potentially very impactful. But I also tell students that on one level was the prize, the privilege of seeing that result and understanding something that hadn't been understood before, the gratification that came with knowing that through our hard work and occasionally reasonably good experiments, we had gotten to this answer. That was the real prize. And so I tried to then avoid getting swept up in the hype of, well, are you going to win the Nobel Prize next year? As it turned out, we did win a number of these sort of pre-Nobel Prizes. And so we, of course, at that point, we thought maybe we were in the conversation, but it's still sort of a surreal experience when you actually get the phone call. I told Thomas Perlman, who I think currently makes the phone calls, he called me, that after Santa Claus, he has the best job in the world, because how can you make anyone any happier than calling them up early in the morning and telling them they've won the Nobel Prize? And at that point, of course, you stop and reflect on, again, how really lucky you've been and what a privilege it's been to have this career. And I'm trying to not have it change my career, other than to now use this as a force for good. And so and people listen to me in a way that they perhaps didn't always listen to me when it comes to, for example, policy matters. And I'm trying to make sure I use my my soapbox to do some good in the world.
Thank you very much indeed. And you're still happy.
Yes.
That's been absolutely marvelous. Thank you so very much indeed. I also want to thank the audience for being with us online and in the auditorium, for the wonderful questions that have come in. It's been gorgeous. I love the thought of all the students going home this evening and doing the closet test, going into their closet and calling out their question and seeing how it reverberates around the closet walls. It's good advice. Well, I think you and I can see a little view of the auditorium. I don't know whether.
Yes.
Yes. So we're waving to the auditorium. It's nice to see you all. Hopefully, yeah, there's some waving back. Fantastic. I don't know whether that's going out to the world at large, but how wonderful to see you. Thank you very much indeed.
Yes.
Bill Kaelin, thank you.